were mRNA vaccines rushed? Today, we're going to look at the history of mRNA vaccines that made the Pfizer and BioNTech and Moderna vaccines possible. First, it might be a good idea to understand what mRNA is. Both DNA and RNA have existed since the beginning of life, and in fact, they're what makes life possible. DNA stands for deoxyribonucleic acid, and it codes for all of the information in our body. It can be thought of as a hard drive or a blueprint. mRNA, on the other hand, communicates all the information from DNA to make the proteins in our cells. You can think of it as the software. That's why it's called mRNA. M stands for messenger, and RNA stands for ribonucleic acid. Here's the fun fact. RNA is made up of codes just like software, but usually on a computer you have 0, 1, 0, 0, 1, and with RNA, the code is made up of letters like A, U, G, C. Now, if we can introduce RNA that we've made ourselves, we can get our cells to make proteins on command to fight an infection or a disease. This is how the current mRNA vaccines work. The very first study with RNA using it for the purpose of a therapy was in 1971 in the UK. It was conducted on rabbits and frogs. They took RNA from rabbit cells and put it into frog cells. This was huge because at this point it was the first time we realized we could make protein from the rabbit RNA in the cells of frogs. But there was a problem, and the problem was that we couldn't do it in humans yet. You see, RNA looks like this, which means it's more degradable from the edges and a lot more fragile than DNA, versus plasmid DNA, which doesn't have an exposed end. Anyways, nobody took the science of RNA seriously because we needed to protect the edges, and the first step towards this came in the 1980s. Actually, it was right here at UBC in the laboratory of Dr. Peter Cullis. He and his team developed lipid nanoparticles, which is actually what we use to protect RNA in the vaccines today. They weren't planning on making it work for RNA specifically, but it definitely laid the groundwork. If we simply inserted RNA into the body, it would get degraded. It's a large, negatively charged molecule, so your cells don't just take it up spontaneously. We have to package it, and so we package it into a lipid particle. Think of it like cheese. You can't just transport it like this. You need it in some sort of packaging until you get it to the customer. In the 1990s, Dr. Katalin Karikó was trying to get RNA to work, and after 5 years of no success, she was demoted from her position at University of Pennsylvania. It wasn't until 2005 when she and her collaborator, Dr. Drew Weissman, were able to figure out how to make RNA work. Remember how I said that RNA is like a code? The solution was figuring out how to change one of the molecular blocks of RNA. It's like changing a tire, scientifically speaking. In 2007, Dr. Derek Rossi, a stem cell biologist, started a lab at Harvard Medical School to build on this work. This led to the creation of Moderna in 2010. Around this same time, Drs. Peter Cullis, Drew Weissman, and Kati Karikó started working on formulating RNA vaccines with lipids. These collaborations eventually led to the work with Pfizer and BioNTech. In 2014, Dr. Kizzy Corbett, a researcher at the NIH, started working on a vaccine for coronavirus. At this point, we had already had 2 coronavirus outbreaks, SARS in 2003 and MERS in 2012. Fast forward to 2019, and we had the outbreak of a coronavirus, which we now know as COVID-19. At this time, all these researchers were working on different diseases, but the whole world turned to COVID-19. Alright, that was a long history lesson, but that's because the history of RNA vaccines is a long one. And its breakthrough worked out just in time, thanks to many researchers along the way. What's amazing about mRNA is that it's easily modifiable as opposed to traditional vaccines. If you want to learn more about this, maybe I'll do another video. And if you're still here,